Here we're going to look at how we can use data from experimental crosses to calculate whether there is genetic linkage between two pairs of genes. In this fruit fly example, normal wings, like this individual here, is dominant over vestigial wings, like the wings this individual here has. And gray body, like this fly, is dominant to black body, like this individual here has. So this fly here has both dominant traits, it's expressing both dominant traits, while this fly over here is expressing both recessive traits. Now we cross these two fruit flies, and this is the offspring that we get from that cross. The question is, what are the genotypes that these flies are producing? Now they produce these 4 types of flies, and there are several hundreds of offspring that were produced in each of these categories. So we have a lot of offspring that were produced from this cross, and they have all of these 4 types of traits. So what do you think the genotype of the parents is? What is the genotype of these flies here? Well, we know that this fly is expressing all the recessive traits, so that means this fly must be homozygous recessive for both traits. So it must be homozygous recessive for the body color allele gene, and we're going to use the letter B to signify body color, and B plus is going to signify the dominant allele here. So this fly, since it's expressing the dominant trait, it must have at least one dominant allele, which we're going to name B plus, and it also has normal wings, which we're going to call VG plus, and that is the dominant allele for the wing structure. While this fly over here is expressing the recessive color, so this fly has 2 recessive alleles, which we're going to label B, and it also has vestigial wings, which is the recessive trait. So it is homozygous recessive for VG. We're going to use the plus for dominant, and without the plus, it will be the recessive allele. But how do we know if this fly over here is homozygous dominant or heterozygous for each of these traits? And we need to look at the offspring. So if we look at the offspring from this cross, we find that some flies have black bodies. So these 2 groups of offspring have black bodies. That means that they have to be homozygous recessive for black body color. For this offspring to have 2 recessive alleles for body color, they receive one of these alleles from the recessive parent over here, but the other allele had to come from this parent. Remember that the offspring has 2 alleles. Each allele came from one of the parents. So this parent must be heterozygous for black body. The same thing, if we look at the offspring with vestigial wings, we see that there are 2 groups of offspring that have vestigial wings. Since vestigial wings is recessive, those offspring must have 2 recessive alleles for vestigial wings. One of these recessive alleles came from the parent that is homozygous recessive, but the other allele had to come from the parent that must be heterozygous. So this parent over here must have also a recessive allele, and that's how this offspring can have 2 recessive alleles. So we conclude that this parent, although it's showing the dominant phenotype, must be heterozygous, and that's how this parent is able to have offspring that are homozygous recessive for each of these traits. Now let's look at what type of gametes each of these parents can produce. So let's assume this fly is a male fly, and it's homozygous recessive for both traits. And of these 2 alleles, only one, because of the principle of segregation, only one will go in the gametes when this fly is producing its sperm. The sperm, instead of having 2 B alleles like every cell in the body of this fly does, the sperm will only have 1 B. And of the 2 VG alleles that this fly has, that every cell in its body has 2 VG alleles, when it is making sperm to reproduce, the sperm will only have 1 VG allele. For the heterozygous parent, it gets a little bit more interesting because it has 2 versions for each gene. So this fly here can produce 4 different types of gametes. Some of those gametes will have the B plus allele, the dominant allele, and another set of gametes are going to have the B allele. Since this fly is also heterozygous for VG, you can have gametes that have the B plus allele and the VG plus allele, or the B plus allele and the recessive VG, or they can have the recessive B with the dominant VG plus allele, or the recessive B allele with the recessive VG allele. So we have 4 different combinations of gametes that this heterozygous fly could be producing. So on the somatic cells, or the cells in the body of this fly, all the cells in the body of this fly have both one dominant allele for B, plus, and a recessive allele for B, and a dominant allele for VG plus allele, and a recessive allele for VG. But when this fly is making gametes to reproduce, so in the ovaries inside of this fly, when it is producing eggs in order to lay those eggs so that they can get fertilized by this other fly, the eggs that this fly is producing would only have one of the versions of the B gene and one of the versions of the VG gene. In which ratios do you expect the gametes will be produced for each of these flies? This fly over here only has one type of allele for both of its chromosomes. It only has the recessive allele, and for VG, both of its chromosomes only have the recessive allele. So we expect that 100% of the gametes this fly produces are going to have a recessive B and a recessive VG allele. But here, since we have 4 possible combinations, we expect that each of these combinations is equally as likely. So a quarter of the gametes produced will have both dominant alleles. A quarter will have the dominant allele for B and the recessive for VG. A quarter the recessive for B and the dominant for VG. And another quarter the recessive for both of these genes. And these gamete ratios then determine what are the offspring ratios that we expect. So now let's cross these 2 flies. What are the phenotype ratios that we expect for the offspring? Let's first look at what offspring do we expect these 2 flies to produce. And here we put a Punnett square where we place the gametes that one of the parents is producing. So we're putting the gametes from this heterozygous female fruit fly, we're putting them on this side, and the gametes that these homozygous recessive fruit fly is producing, we're putting them on this side over here. And now we're going to look what would be the resulting offspring if this gamete fertilizes this gamete over here, or if this gamete fertilizes this gamete over here, and again, if this gamete fertilizes this, or if this gamete fertilizes this. So those are the 4 possible gamete combinations once this fruit fly lays its eggs and this other fruit fly puts the sperm to fertilize those eggs. That what are the possible combinations that could happen. And here we have the possible offspring that will be formed from those combinations of gametes. At what ratios do you expect each of these genotype and phenotype combinations? In this case, each genotype has also a distinct phenotype. So in this case, the genotype and the phenotype ratios will be the same. But it's not always the case, since homozygous dominant have the same phenotype as heterozygous, but they don't have the same genotype. But we don't have any homozygous dominant in this offspring. And since we have predicted that all of these gametes will be formed in the same ratios, we expect that all of these offspring will also be in equal ratios, so that 25% or a quarter of the offspring will have normal wings and gray bodies, 25% or a quarter of the offspring will have black bodies and vestigial wings, 25% will have gray bodies and vestigial wings, and 25% will have black bodies and normal wings. What are we assuming when we calculate these ratios? What are we assuming when we say all of these gametes are equally as likely to form? And we are assuming that the genes, these 2 genes, the gene for body color and the gene for wing structure, are assorted independently. And there are 2 ways in which genes assort independently. It could be that the genes are located on different chromosomes, like for example this E and D gene. The E gene is located on this small chromosome. Here we have a cell, and this cell has 3 pairs of chromosomes, and is heterozygous for all of these genes. We have a small chromosome, we have 2 versions of that small chromosome, a medium chromosome and a large chromosome. So if the genes are located on different chromosomes, like the E gene, which is located on the small chromosome, and the D gene, which is located on the medium chromosome, those two genes would assort independently because they're located on different chromosomes. Genes could also assort independently if they are located on the same chromosome but very far apart. So this F gene and the C gene are located in the long chromosome, the C and the F gene, and since they're so far apart, crossing over is very likely to happen in between them. When this individual is doing meiosis, the gametes it produces could have this dominant F from this chromosome, and with crossing over, it can be on the same chromosome in the gamete as the recessive C allele from the homologous chromosome. Genes that are located on different chromosomes or on the same chromosome but very far apart would assort independently. On the other hand, genes that are located on the same chromosome but very close together, like here C, B, and A genes, these three genes are so close together that the chances of crossing over happening between them are very low, so this combination is more likely to be inherited and passed on in the gametes, and this combination than having a mix of this dominant C with the recessive B and the dominant A. That is an unlikely combination because crossing over will need to happen exactly in between these two genes for those combinations to appear. So when we mate these two flies, they produce hundreds and hundreds of offspring. So we're looking at actually thousands of offspring that they produce. And this is very important that we're looking at a large number of offspring because this will give us more confidence in the assertions that we make. If you were doing this experiment in humans, for example, and you're looking at the gene for having a widow's peak with your hair pattern and disliking broccoli, like the ability to taste PTC, and the crossing that you are looking at only has 4 offspring, and let's say 2 of those kids dislike broccoli and have a widow's peak, and the other 2 kids like broccoli and don't have a widow's peak, that is not enough for you to say that those 2 genes are not assorted independently because you're only looking at 4 offspring. It's a very small number and you could have gotten that result just by chance. So for example, if you're flipping a coin, you might be able to flip 2 coins at the same time and get heads in both coins. That doesn't mean that your coins are linked. That could just happen by luck. Now, if you flip your coins 100 times and both times you get the same result in both coins, then you can start saying, okay, something is happening between these 2 coins. They might be linked. The same thing is here. Since we have thousands of offspring, now we can make some good conclusions about what's happening here. And notice for this first type of offspring, we have 965. For this type of offspring, we have 944. This type of offspring, 206. And this one, 185 of the fruit flies came out looking like this. So the thousands of babies that these 2 parents produce, these are the ratios of the observed phenotypes. Based on these ratios, can you assume that the genes for body color and wing type are assorted independently? And let's go back to what we had calculated before. We had said if these 2 genes assort independently, we would expect a 1:1:1:1 ratio of all the offspring possible phenotypes produced. So we would have expected that the ratios of these offspring would have been pretty similar. So if we have 2,000 over 2,000 offspring, you would expect that about 500 or so will be in each of these categories. It won't be exact 500 each, but somewhere around there. Instead, what we have is almost 4 or 5 times more of these offspring than on this offspring. So what do the observed offspring ratios tell us about the location of these 2 genes in the chromosome? So if these 2 genes were assorted independently, we could say that they're either located on different chromosomes or that they're located on the same chromosome but very far apart. But since these 2 genes are not assorted independently, some combinations are more likely than others, we can say that these two genes must be located very close together on the same chromosome. So they're on the same chromosome and very close to each other. And now we can use this information from the offspring to figure out what is the configuration of these alleles in the chromosomes of the parents. So we can look at this parent fruit flies and figure out what are the chromosomes in their cells, in all the rest of their cells, what do they look like? Let's start with the homozygous fly that is homozygous for both genes. Let's assume that the B gene is located here at the top. This is the locus for this gene, is the section of DNA at the top of the chromosome. Since this fly is homozygous, both chromosomes, homologous chromosomes, will have the recessive allele located at the top in that section of DNA at the top of the chromosome. For this fly is heterozygous for the B gene. So one of its chromosomes is going to have the dominant allele and the homologous chromosome is going to have the recessive allele. For BG, since this fly is homozygous, both chromosomes, both homologs, are going to have the VG allele. And for this fly, it could have either the VG plus or the VG. But how do we know if the VG plus goes here with the V plus or if the VG plus goes here with the recessive V? And for this, we need to look at the possible gametes that this fruit fly could produce and which of those gametes were more common than the others. This fly here only produces one type of gamete since it's homozygous for the B gene and homozygous for the V gene. All the gametes are going to be the same because it only has the same type of allele in both chromosomes. What we find is that these two types of offspring are the most common. So like these two types of offspring are 4 to 5 times more common than the other type of offspring. So that means that this type of gametes that this heterozygous fly is producing are a lot more common. So this fly is producing a lot more of these gametes than it is producing this type of gametes. So what that means is that when the gametes that have the B plus allele are more likely to also have the VG plus allele, and very few gametes have the B plus allele with a recessive VG. On the other hand, the gametes that have the recessive B allele also have the recessive VG allele, and very few of the gametes have the recessive B allele with the dominant VG plus allele. Now, this is not because the dominant like to go with dominant or recessive like to go with recessive. This has to go with what is the location of those alleles on the original chromosome in this fruit fly. And based on what is the location of those alleles in this chromosome, those in the original combination will be more likely to happen than those that will require a crossing over event in order to form. So that tells us since the B plus was most likely to be inherited with the VG plus, they have to be on the same chromosome, while the homolog will have the B and the VG, the recessive V with the recessive VG allele, because most of the gametes that were formed that had the recessive V allele also have the recessive VG allele. And most of the gametes that have the B plus allele also have the dominant VG plus allele. And very few had the other possibility. These gametes that were most common, they just had the same original setup as the parental chromosomes in this fruit fly. So since one of the chromosomes in this fruit fly has both dominant alleles and the other chromosome has both recessive alleles, most of the gametes will have both dominant alleles in one side and both recessive alleles on the other side. And the few gametes that have one of the dominant alleles with the other recessive allele resulted when crossing over happened right in between these two genes. So again, most of the time crossing over happened somewhere else. So there was crossing over here, but it just didn't change the order of the B plus next to the VG plus. Only when crossing over happened right in between these two genes, you form gametes like this that had a B plus from one of the chromosomes with the VG from the other homolog. And the same for here. So in those few occasions where crossing over happened right in between those two genes, you have this that are called the recombinant combinations. If this fly instead had had the B plus with the recessive VG over here and the B with the dominant VG over here, then you would have seen that those are the ones that are more common. So we see most of the gametes that this fruit fly is producing will have the original combination as it's in its original chromosomes. And the few gametes that have a different combination are from the rare cases in which crossing over happened right in between these two genes, the B and the BG allele. And when crossing over happened between those two genes, now you have recombinant combinations where you have the B plus allele going with the recessive VG or the recessive D going with the dominant VG plus allele. And the question now is, looking at this fruit fly, what combination of alleles did this fruit fly, this parent fruit fly, get from its own parents? Since one of the chromosomes has both recessive alleles and its other chromosome have both dominant alleles, one of these chromosomes, this fruit fly inherited from one of its parents. So one of these chromosomes came in the egg of this that the mother of this fruit fly made, and the other chromosome came in the sperm that the dad of this fruit fly gave it. One of the parents gave both recessive alleles to this fly, and the other parent gave both dominant alleles to this fly. And remember, the chromosomes that we get from our mother and the chromosome we get from our father, they are homologs and they stay in the same configuration as we got them from each of our parents in all the cells of our body. And the only time that those chromosomes undergo recombination, in the only place they do that is in the gonads, in the space of the gonads where the sex cells are being produced. Only when the sperm and the egg are being produced. And the only cells that are mixed, that have a mix of our mom and our dad's chromosomes, are the sex cells that we're going to pass on to our offspring. So only the eggs and the sperm will have combinations from our mom and our dad. But the cells in our body will just have the chromosomes exactly like we got it from our mom and exactly as we got them from our dad.